In this video, we will discuss about this chemoresistance in the pancreatic cancer cell is reversible and is dependent on the stiffness. So far, we have discussed that in the high help matrices, which composed of HA proteins and elastin-like proteins, and under more stiffer conditions, it will cause this chemoresistance through the HA interactions with the CD44, and that leads to the upregulations of this drug efflux transporter ABCG2. That things we have discussed in our previous videos. You can refer to those videos to understand this relationship. In this video, we will discuss whether this chemoresistance that is developed by high help matrices is reversible when the cells or organoid are switched back to the low mediated chemoresistance, low mediated or low stiffness, low help matrices. Yeah, that's the right word. So we will discuss that thing. The hypothesis that they comes to the mind of the researchers is based on the fact that in the low help matrices, even though the drug transport is similar to the high help matrices, we see the low chemoresistance in the pancreatic cancer cells compared to the case of high help matrices. So that's the basis of the hypothesis, and they want to check this hypothesis. So in the panel A, they have discussed the approach that they have followed for this experiment is that first they grow the cells and do the drug treatment at the passage 1. After that, what they did is they interchange the matrices at passage 4 and then continue to grow these cells till passage 7 before they measure this chemoresistance. And one thing that they have observed, and that is also expected, is that once they switch the matrices of the organoid that are developed in their respective matrices, they can see that to some extent the organoid still is able to remember or still able to memorize its previous matrices conditions to some extent, and these things will be reduced after the third passage. And that can be seen in this panel C, where we can see that the dotted line indicate the transition that occurs, whereas if this transition would not have occurred, what would be the IC50 that is shown here, and similar for the case of this low help matrices. And these things in the panel C, it is shown here for the single cells, whereas in the panel D, it is shown for the organoid, and the observations are similar. That is, once they switch the matrices, cells are able to remember or memorize its previous matrices conditions to some extent. And however, after passage 7, they can see that this they are able to adapt themselves to the new matrices. And when they check this cell viability in the single cells at passage 7, after passage 7, they can see that there is a difference between cell viability in the high help matrices and the low help matrices. In the low help matrices, they can see that cell viabilities they can in the high help matrices, they can see that the GC150 requirements is high for the same cell viability compared to the this low help matrices. And same things they have observed after and these things are switched when they and these things are reversed when they switch the cells into the different matrices. That is, in the when they switch the cells from low matrices to the high matrices, they can see that the IC50 requirement are increased, indicating that the chemoresistance in the switch from low to high matrices cause more this when the organelle or cells, in this case when the cells switched from low to high matrices, it will cause more chemoresistance. However, when the cells are switched from high help to low help, they can see that reductions in the gemcitabine IC50 for the same cell viability, indicating that chemoresistance reduced when the cells are switched from high help matrices to the low help matrices. And same things they have observed for the organoid, that is when there is no switch, they see that the high help matrices has more cells toxicity at the particular gemcitabine concentrations compared to the low help matrices, and these things are reversed when the cells are switched from low to high help matrices, where we can see that cells of toxicity for the same gemcitabine concentration is high for the switch from from the help low matrices to the high help matrices. And after confirming their hypothesis that when the cells are switched from different matrices, this chemoresistance can be reversed, or when the cells are switched from stiff matrices to the soft matrices, this chemoresistance can be reduced. And these things are quantified in the panel F, where not these things, the other things that cause this chemoresistance are also quantified in the panel F, where we can see that the CD44, this CD44 is kind of low in the case of help low matrices, and also when the transition occurs from high help to low help matrices, whereas the CD44 is high in the help high matrices, or when the transitions occurs from help low to help high matrices, indicating that in the high stiff matrices there is more CD44 productions, and that will cause this more ABCG2, that is drug efflux transport upregulations, and this results in the increased chemoresistance against the drugs that are used to target cancer cells. And this is the final overview that they have hypothesis, or they have predicted, or they have concluded from their experiments, that is in the soft extracellular matrix, the drug efflux pumps is downregulated and chemoresistance are also downregulated, whereas in the case of stiff ECM or stiff extracellular matrix, the CD44 and HA signaling increases, that will cause drug efflux pumps increases, that is ABCG2 genes, and that will also cause increase in the chemoresistance. This is what the final summary of the paper, this panel G. If you need to understand these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.